explaining. Is it clear?
Yes, ma'am.
Hey guys, and welcome to Hari Astro. In today's video, we're going to be talking about a very interesting topic, and that is Amibiasis, which is also commonly known as amoebic dysentery. So let's get started. So what is Amibiasis? Amibiasis is a parasitic infection of the intestines which is caused by a parasite called Entameoba histolytica. This parasite can affect anyone, although it is most common in people who live in tropical areas with poor sanitary conditions. The disease is actually commonly asymptomatic, but symptoms ranging from mild diarrhea to severe dysentery may occur. Some extraintestinal infections can include a liver abscess, and worldwide each year, an estimated 40 to 50 million people develop amoebic colitis or extraintestinal disease, and about 40 to 70,000 people die. So from this definition of amoebiasis, we get that it's a parasitic infection which is caused by this parasite called Entameoba histolytica. So when this parasite enters the body, it actually infects and affects the GI tract centrally, and usually the intestines, and it gives the patient abdominal pain and cramping as well as diarrhea. And it's usually common in tropical areas where we have poor sanitary conditions. So the disease can also be asymptomatic in many patients, but some patients may develop a mild diarrhea, or some may even go on to develop a severe dysentery. So in the majority of cases, the Entameoba histolytica parasite actually infiltrates just the intestines in the GI tract. But sometimes we can have extraintestinal infections in more severe cases of infection. And they can also go on to cause liver abscesses by infiltrating the liver, or even an amoebic colitis, which causes an extreme amoebic dysentery for the patient. So now that we know what the basics of amoebiasis is, let's take a closer look at how one can contract this disease. So the parasite actually lives only in humans and is passed in the feces, which is the poop of the infected person. So a person gets amoebiasis by eating fruits and veggies that has touched infected feces, or by eating or drinking food or water which is contaminated with the parasite. It can also be spread sexually by people during oral anal contact or oral anal sexual activities. And an interesting point to bring up is that some people with amoebiasis may carry this parasite on for weeks to years, often without symptoms, but continuously passing them into their feces. So if we take a closer look at this image to the right of my screen, it shows us the most common way in which the disease is actually spread. So we have these cysts and trophozoites which are passed into the soil from the human feces, and this feces can go on to contaminate water sources which are used to irrigate plants or veggies and fruits, or the human can actually get it by also ingesting this contaminated water. So the cysts are ingested in contaminated food or water, and then we have the excistation, which occurs in the small intestines, and this process releases single motile trophozoites of this parasite that colonize the large bowel. So we have the excistation process in the small intestine, and then we have these motile trophozoites which colonize the large intestine. And as we mentioned earlier, 90% or 9 out of 10 patients will actually remain asymptomatic with these trophozoites in their large intestine. But that doesn't mean that they still can pass the infection onto others by contaminating food or water sources. But in the patients who do develop symptoms, the trophozoites can invade the large bowel, causing flask-like shaped ulcers and bloody diarrhea for these patients. And from there, we can have the extraintestinal manifestations, as we said. So following the GI infection, which is usually asymptomatic, these parasitic trophozoites may invade the liver through the bloodstream, causing necrotic abscesses And this is how the cycle continues, and this is how the disease goes on to be spread. So an interesting fact that's not actually brought up in this picture is that the disease can also be spread by oral anal contact. So any sort of oral anal sex will also cause a person to ingest these cysts from the anal contact with the infected individual. So now let's talk about some signs and symptoms of the disease. So on average, about 1 in 10 patients who are infected with the Entameoba histolytica parasite become sick from the infection, which means that the vast majority of these patients will be asymptomatic. So the symptoms these patients suffer from include loose stools, stomach pain, flatulence, fatigue, and stomach cramping. So amoebic dysentery is a severe form of amoebiasis and is associated with stomach pain, bloody stools, and a fever. And in some rare cases, the Entameoba histolytica parasite will invade the liver to form an abscess, and in even less common cases, it may spread to other parts of the patient's body, such as their lungs or brain, and cause abscesses even here. The diagnosis of amoebiasis. So the examination of the stools, which is the poop, under the microscope is the most common way to diagnose amoebiasis. So sometimes several stool samples must be obtained because the number of amoeba being passed in the stool varies from day to day and may be too low to detect from any single sample. So if we take a closer look at these two microscopic images at the bottom of my screen, the first one is actually a trichome stain of Entameoba histolytica, and we can see what these parasitic trophozoites look like. And this is actually the typical aspect of the Entameoba histolytica trophozoite on microscopy. And finally, let's talk about the treatment of amoebiasis. So the treatment of this disease includes metronidazole and tinidazole, and metronidazole is usually given for 10 days either by mouth or intravenously. And to kill the amoebas and cysts confined to the intestine, 3 drugs called luminal drugs may be administered. They include idoquinol, paromomycin, and diloxinate furoate, which can help to kill the amoebas and cysts within the intestine. And that brings us to the end of this video on amoebiasis. Thank you guys so much for watching. Hope you
I hope you got an idea, students.
Yes, ma'am.
Yes, ma'am.
So now we will look into details. So coming to the taxonomy, the organism belongs to the kingdom Protista, phylum Sarcomastigophora, subphylum Sarcodina, and superclass Rhizopoda, and class Lobosa, and genus Entamoeba. There are various species included under this. We will just go in detail about the Entamoeba histolytica. So coming to the introduction part, the genus Entamoeba was defined by the scientists known as Casagrande and Barbagallo in the year 1895, and this term, the word Entamoeba, was derived from the Greek words like entos means within and amoeba means change. And this Entamoeba histolytica is considered as the common protozoan parasite which will be found in large intestine of humans. So it will colonize in large intestine of humans, and this organism causes the diseases known as Amibiasis as well as liver abscess. So this is most common disease caused by these organisms, but if it is not treated well, this may also lead to liver abscess, and also it can damage the other organs. And this infection is caused, the parasite is caused at a 3rd leading parasite cause of death in the developing countries. And Entamoeba histolytica was first described by the scientist known as Lamble in year 1859 in the colonic autopsy of a child who died of diarrhea. And the scientist known as Cartulis confirmed that amoeba is the causative agent for the diseases called as Amibiasis, or you can call it as amoebic dysentery, as well as the liver abscess. So he has confirmed that amoeba is responsible for causing these diseases. And later on, Feder Loschin described the pathogenic nature of Entamoeba histolytica. That means how it is able to cause the disease. That was proven by the Loschin. So where he had inoculated the parasite through the rectum of a dog and found that the dog had developed this dysentery or amoebiasis, thus proving the pathogenic nature of the parasite. So in the previous slide, I had said that it is the intestinal parasite. So it is residing in the large intestine and causing the disease. So the route of entry and how it is infecting, what morphological changes it is undergoing, all those has been described by the Loschin when he is studying about the pathogenic nature, and detailed life cycle was studied by the scientist known as Dobel in the year 1925. These are not so important. Whatever you can remember, those scientists' name you can. Now coming to the epidemiology, as I said, we had seen in the first slide, this is the third leading parasitic infection in the developing countries like Bangladesh, India, Pakistan and Nepal, and about 500 million people are infected every year, of which 50 million suffers from the disease and 100,000 people will die due to this amoebiasis as well as liver abscess. And it is very common in adults, rare in children, and amoebic liver abscess is more common in males because of their lifestyle. The males are more prone to these infections. Now coming to the details about the morphology. So these parasites occurs in 3 stages. What are those 3 stages? Trophozoite, pre-cyst and cyst. These are the 3 important morphological forms of this parasite. So just what are these 3 morphological forms means? The trophozoite is also known as feeding stage or free moving stage, which lives in the mucosa and submucosa of large intestine, especially the colon and the cecum. Here you can see the picture, colon and the cecum. So in this area, the trophozoite will be colonizing. And pre-cyst, it is a transient stage between the trophozoite and the cyst, which is formed in the lumen of the large intestine. So this will be present in the colon and cecum, whereas pre-cyst will be residing in the lumen of the large intestine. And the cyst, it is a non-feeding infective stage of the parasite found again in the lumen of the large intestine. So this is general about, a rough idea about what are these different morphological forms of the parasite. Now coming in detail about each form, the first one, trophozoite. If you look at the size, it varies in size about 8 to 40 micrometers, but it can go up to 20 to 25 micrometers in size. And the shape is not fixed, constantly changing its shape by thrusting out pseudopodia. As it consists of pseudopodia, it doesn't have any definite shape. Now coming to the cytoplasm, the cytoplasm is differentiated into clear, thin, translucent ectoplasm. You can see it in the diagram here. This is the thin, translucent ectoplasm, and a central fluid like granular endoplasm. This endoplasm contains the nucleus, numerous food vacuoles, as well as erythrocytes, RBC cells, and granules and tissue debris. And this trophozoite, this is important point. Please remember, trophozoite is an anaerobic parasite. It lacks mitochondria, Golgi bodies, and rough endoplasmic reticulum. So these 3 membrane-bound organelles are absent in this trophozoite form because this is considered as the anaerobic parasite. It is not undergoing the oxidative phosphorylation. So that's why it doesn't require these organelles to be there in the cytoplasm. So that's why it is devoid of these cell organelles. So you need to be thorough with the diagram. Please practice the diagram. Coming to the nucleus part of this trophozoite, it is very peculiar for structure it attains. What is it means? The nucleus, as is evident from the figure given below, nucleus is the most distinguished feature of this Entamoeba histolytica trophozoite form, where it spherical in shape and the size ranges up to 3.5 micrometers and lined externally by a thin, delicate nuclear membrane. This is considered as a nuclear membrane, the gray color. So thin delicate nuclear membrane will be enclosed, and this nuclear membrane is lined internally. You can see the dark black line here, lined internally by a single layer of evenly distributed chromatin granules. These are considered as the chromatin granules. And these chromatin granules are present in the form of dots, but as they have been joined, you can see as a thick black color line over here. So the chromatin granules are in the form of small dots. A compact, another important component is a compact small karyosome or endosome surrounded by a clear halo ring is centrally located. So this is the endosome around which the rings will be present, and it nuclear striations or spoke-like lines will be radiate out from the endosome and extend till the nuclear membrane like wheel. If you look at the wheel or our national flag, at the center we'll just put the Ashoka chakra. So in the same way, you can see the shape of the the structure of the nucleus inside this trophozoite form. Okay, and under the microscope you can see the the trophozoite in this form. Okay, so cyst, you can see the pseudopodia here, and these trophozoites are exclusively parasitic forms. So these are responsible for causing the disease. You need to be thorough with the diagram, students. Now coming to the another morphological form, that is pre-cyst, which is smaller in size, about 10 to 20 micrometers than trophozoite, but larger than cyst. If you look at the shape, it is round or oval with a blunt pseudopodia. Very small pseudopodia will be present when you compare with the trophozoite stage. It has a relatively larger nucleus that retains all the characteristics of the nucleus of a trophozoite, so which is similar to the trophozoite. The trophozoite extrudes food vacuoles before encystment so that the endoplasm is free from red blood cells and other ingested food particles. So you can't see any food vacuoles or erythrocytes or any other organelles. So all those will be excluded to when the trophozoite is getting transferred into the pre-cyst. Only you can see the presence of the nucleus inside the cytoplasm. Now coming to the another important morphological form, that is cyst. So again, the cyst can exist in three forms. Those are immature uninucleate cyst, binucleate cyst, and mature quadrinucleate cyst. That means it consists of only one nuclei, here two nuclei, here again it will undergo the nuclear division to form the fourth. So that stage is very infective. This is the cyst which will be transformed from one host cell to the another host cell. But the parasitic or infective stage is trophozoite only, but cyst will be transferred from one person to the another person through stool sample. The trophozoite becomes completely round and surrounded by a transparent, highly retractile double-wall resistant cyst wall during the encystation process, and the cytoplasm is clear and transparent. Before encystment, the parasite eliminates food vacuoles and accumulates considerable amount of food in the form of glycogen mass and black bodies called as the chromatoidal bars or the bodies. So when trophozoite is getting transferred to the cyst, so all the food vacuoles and will be concentrated to form these black color mass bodies, which are called as the chromatoidal bars or chromatoidal bodies. So these bodies are large, smooth, oblong rods with rounded ends, and they may be 1 to several in number. They are called as the chromatoidal bars of the bodies because they stain. This is important because they stain as chromatin with hematoxylin stain. So when we are staining this cyst, we can stain them by using this particular stain. It is believed that the chromatoidal bodies contain DNA and phosphates and serve as a storage of the ribosomes. So apart from storage of the glycogen, they are also considered as the storage of the DNA material along with other phosphates and ribosomes. Cyst when formed is uninucleate, but the nucleus within the cyst divides twice to form quadrinucleate cyst. It is this mature quadrinucleate cyst which is the infective stage. So you need to be thorough again with the diagram. So if you know the diagram, by looking at that, you can describe it. So this one is the uninucleate cyst because it consists of only single nucleus, whereas this is the Matured quadrinucleate cyst. So here you can see the 4 cysts, and this is the microscopic observation. Coming to the life cycle, so the host is a man, and the infective stage is the mature quadrilateral cyst, and route of infection is fecal or oral route, and site of invasion is large intestine, and diagnostic stage is we can diagnose the presence of cyst or thrombozoids in the sample, and the pathogenic stage is thrombozoid, so which can cause the disease. Now coming to the life cycle, so please write it in the flowchart manner. I'll be giving the steps, so you know how to write the flowchart, right students? Can you do that now? Because it will be easy to remember. I hope everyone has running notes.
Yes, ma'am.
Okay. So under life cycle, the first step, write it as ingestion of contaminated food. Ingestion of contaminated food, within the brackets, mature quadrinucleate cyst. Please make a note. All these are not there in the PPT. I want everybody to write these steps in the flowchart manner. The first step is ingestion of contaminated food, within the brackets, mature quadrinucleate cyst. Put an arrow downwards and go with the second step. Cyst resistant to acidic conditions in stomach. Cyst resistant to acidic conditions in stomach. Therefore passes without any changes. Therefore passes without any changes into small intestine. Okay, next step. Excistation occurs in small intestine. Excistation, EX. Excistation occurs in small intestine due to alkaline conditions. Are you able to write it, ma'am?
Yes, ma'am. Yes, ma'am.
Okay. Now again put an arrow downwards. Next step. During this process, that means during excistation, cytoplasmic body retracts, retracts and loosen from the cyst wall, that means cytoplasm will contract and it will loses from the cyst wall. It won't bind firmly to the cyst wall. Slightly there will be a contraction in the cytoplasm, and cyst wall will the connection between the cytoplasm and cyst wall will be destroyed here. Cytoplasmic body retracts and loosens from the cyst wall, pseudopodia is formed. That means slowly the cyst is entering into the thrombozoid stage here. Okay, pseudopodia is formed.
Ma'am, can you repeat again?
During this process, cytoplasmic body retracts and loosen from cyst wall, and loosen, LOOSEN, and loosen from cyst wall, pseudopodia is formed. Next step. As a result, Tetranucleate amoeba. Tetranucleate amoeba known as metacyst is formed. Tetranucleate amoeba known as metacyst is formed. Meta, M-E-T-A. Metacyst is formed. Okay. Next point. Nuclear division occurs. Nuclear division occurs within the brackets. 4 gives rise to 8. You just put an arrow. 4. That means 4 nuclei are becoming 8 nuclei now. Are you getting my point, ma'am?
Yes, ma'am.
Yes.
Just write it as 4 giving rise to 8 nuclei within the brackets. Now coming to the next step, 8 amebula are formed. So as a result of 8 nuclei has been there, each nuclei will surrounded by the cytoplasm. As a result, 8 amebulae, write it as, the spelling is Amoebulae. Total 8 amebulae will be formed. 8 amebulae are formed. Okay. Next step, put an arrow downwards and write down the next step. Pass through the cecum. Pass through the cecum. C-E-A-C-U-M of the intestine. Pass through the cecum of the intestine. Next step, invade the tissues. Invade, I-N-V-A-D-E. Invade the tissues and lodges in, stays, or you can simply call it as stay in mucosa and submucosa of large intestine. Invade the tissues and stays in mucosa and submucosa of large intestine. Finished? Up to here?
Yes, ma'am.
Now again put an arrow downwards. Next step, trophocytes grow. Trophocytes grow within the brackets by binary fission. Again they will divide. Trophocytes grow. That means in the brackets write it as binary fission. The next step, secrete histolysin. Very, very important. Secrete histolysin. H-I-S-T-O-L-Y-S-I-N. Histolysin. Just write it in the brackets. Necrosis and destruction of host tissue. Necrosis, N-E-C-R-O-S-I-S. Necrosis and destruction of host tissue.
Ma'am, repeat once again.
Secrete histolysin, within the brackets, necrosis and destruction of host tissue. Again, keep an arrow downwards. So when you are just putting an arrow like this, near to the arrow, write it as amoebic dysentery, or simply write it as Amibiasis. So by this, the disease called as Amibiasis will occur. Okay. Now here just put another step. Under unfavorable conditions, in lumen of large intestine, under unfavorable conditions in lumen of large intestine. Okay, again put an arrow like this and write it as develop cyst, develop cyst, and again see here, you had written it as develop cyst na. So within the brackets again a small flowchart will be there here. Don't include into the main flowchart. Write it as develop cyst, and here you just put small flowchart like pre-cyst. Are you getting my point, ma? Pre-cyst.
Yes.
In the brackets this has to come. Pre-cyst gives rise to uninucleate cyst. Uninucleate cyst. Again write it as binucleate cyst. And again write it as quadrinucleate cyst. Okay, quadrinucleate cyst. You had written this small flowchart na, so here just put an draw like this and write it as an existation. Write it as existation here. So this process is known as encystation. Encystation, formation of cyst na. So write it as encystation. So now we are ready with the quadrilateral nucleate cyst here. Now in the main flowchart, just put an arrow like this, and the next step you write it as passes out with feces. Passes out with feces or with stool. Okay, passes out with stool. Again, in the next step, again put an arrow downwards. On the arrow, when you are just putting the arrow like this, beside the arrow write it as in few cases. In few cases, what will happen? Spread to the bloodstream. Here you write down. Spread to the bloodstream and put an arrow. Write it as causes liver abscess. Causes liver abscess. So this is about the life cycle of Entamoeba histolytica. Okay. So what it is doing actually? Yes, ma'am.
Ma'am, after in few cases?
Few cases spreads to bloodstream. Spreads to bloodstream. And next last stage, write it as causes liver abscess. Liver abscess. ABSCESS abscess. That is pus formation will be there. So whenever there is a pus formation or enlargement has taken place, that type of infection will be calling it as abscess. So shall I go through the life cycle once again?
Yes, ma'am.
Okay. So what it is happening during the life cycle, how it is able to cause the disease means, as I said, these thrombozoites, quadrinucleate cyst will be ingested through contaminated food, and these cysts will pass on through the ingestion, and they will pass on to the small intestine, and where the existation process occurs due to the alkaline conditions in the small intestine. And during this existation process, you can see the cytoplasmic body retracts and loosens from the cyst wall. Pseudopodia formation will be there. As a result, so here you can see slowly the cyst has been transferred into the thrombozoid stage. So this type of thrombozoid is known as a metacyst, and this will converted into this is the major infective stage which colonize the large intestine without any symptoms. The host cell can become a carrier throughout the life cycle. Few people are asymptomatic, they won't get any symptoms, so simply they will carry and they will spread the disease from one person to the another person, or it may cause the disease inside the host cell, which results in the amoebic colitis or amoebiasis, or it may spread to the other organs, especially the liver, causing the amoebic liver abscess. Okay, and this metacyst, which is again undergo the nuclear division to form 8 amoebulae, and these 8 amoebulae will pass through the large intestine, and they try to invade the submucosa and mucosal layer of the larger intestine, and they will increase in their number. So total 8 amoebulae has been there in the large intestine. Again, this 8 amoebulae will undergo binary fusion to increase its number, and they start secreting the component known as histolysine. Due to the secretion of this histolysine, it is able to destroy or destruct the submucosa and mucosal layer of large intestine. So as it is destructing the host tissue there, you can see the bloody stools. Blood will be excreted through the stool. So this is not happening in the case of giardiasis. It is there, giardia is just blocking the microvilli action of absorbing the nutrients from the food, but here it is destroying the host cell by producing the histolysis. As a result, you can see the excretion of the blood along with the stool. So that is the main difference between the giardiasis and amoebiasis. In giardiasis, you can see the stool diarrhea condition, but without greasy diarrhea will be there, where fat will be released into the stool, but no blood will be seen. But in this amoebiasis, you can see along with the stool, blood stains also will be there. Okay, why blood has been released? Due to the destruction of the host cell in the when the thrombozoid is invading the mucosa and submucosa of the large intestine. Okay, so it won't end up there. If proper treatment is not given, again this thrombozoid will undergo the encystation process. Encystation process in the larger intestine itself, where the uninucleate cyst will convert into binucleate and quadrinucleate, and then it will passes into the stool along with the blood, or sometimes from here it will get into the bloodstream, and it may cause the liver disease, or it may go to the brain also. It can damage the brain, as well as you can, in few cases you can see the it is infecting the skin also. Skin infections also will be there. So those are the worst or the chronic stages of this disease. Okay, if we identify at this stage only, when amoebiasis or diarrhea symptom has been notified, then we can treat with the medicines. So these liver abscess and damaging the brain and other organs are the chronic stage of this disease. Is it clear, ma? Life cycle.
Yes, ma'am, clear.
So again, I am telling that just go with the life cycle in this flowchart manner only. It will be easy for you people to remember. Yes?
Yes, ma'am.
Shall we see the pathogenesis in the next class then?
Okay, ma'am.
For you people. So I'm just ending up the class here. Yeah, please give me the attendance, ma.